Hi, I'm Peter Libby from Boston's Brigham and Women's Hospital and the Harvard Medical School, and it's my pleasure to be here today. I view residual risk in falling into several different categories. There's residual lipid risk, those who are having events with an LDL above your target level. There's residual glycemic risk, people who have poorly controlled diabetes. There's residual thrombotic risk. The problem there is that we don't have a biomarker that's convenient and scalable to measure it. And then there's the space where I live, which is in residual inflammatory risk. And it actually turns out that the residual inflammatory risk in our well-treated statin populations is actually a greater proportion of our patient population than those with residual LDL risk. So that's why I've devoted a great deal over the last 15, almost 20 years to trying to attack residual inflammatory risk in the clinic. Inflammation as a driver of atherosclerosis is now fairly well accepted, or beginning to be accepted, but when we started this journey, it was very controversial indeed. And together with my clinical trialist partner, Paul Ritker, we've considered a number of possible targets, and we focused on interleukin-1 beta for a number of reasons. One of which is we had a tremendous amount of preclinical data that suggested that it would be a viable target. And also because it was the art of the possible, because there was a very good monoclonal antibody that specifically neutralizes interleukin-1 beta, canakinumab. So we were able to convince our corporate sponsor in a very enlightened series of conversations to embark on the journey of doing a large-scale cardiovascular endpoint trial that enrolled people who had survived a myocardial infarction, were in the chronic phase of ischemic heart disease, who were on a full menu of all of the good things that we do in secondary prevention, certainly when we started the trial in 2011, and in particular had their LDL very well controlled. And in fact, the LDL level in the trial, the canakinumab anti-inflammatory thrombotic outcomes trial, CANTOS, was about 10 milligrams per deciliter lower than in the pivotal PCSK9 trials. And the patients who were enrolled, however, despite being on excellent contemporary therapy, had a little flag waving saying that they had residual inflammation, as gauged by an elevation in the inflammatory biomarker C-reactive protein measured with a high sensitivity assay, greater than 2 milligrams per liter. And in that trial, we found that there was a reduction in hard cardiovascular endpoints at the end of the day. And this was the first trial that established the inflammatory hypothesis in humans and pointed the beginning of a pathway towards anti-inflammatory therapies that may allow us to address this basket of inflammatory residual risk. Well, we're very fortunate that there's a pair of trials with an old tried and true anti-inflammatory medicine, colchicine. We've become familiar in cardiology using colchicine in treating pericarditis, and the investigators of the COLCOT trial and of the LODOCO-2 trial took two different populations, one soon after an acute coronary syndrome and the other in the chronic phase, more chronic phase of ischemic heart disease, randomized them to low-dose colchicine, and was able to show striking reductions in events. So this is a wonderful affirmation of the inflammation hypothesis and gives us a drug that we can take off the shelf today and treat our patients that we consider meet the eligibility criteria for those trials and in whom we think an anti-inflammatory agent would be of benefit.